The overall goal of the following procedure is to rapidly generate donor-derived virus-specific cytotoxic T lymphocytes, or CTLs, that are suitable for adoptive transfer to allogeneic hematopoietic stem cell transplant, or HSCT, recipients. This is achieved by first nucleoaffecting dendritic cells, or DCs, with plasmids encoding viral antigens to prime the cells for antigen presentation. As a second step, the nucleoaffected DCs are used to stimulate antigen-specific T cells, which are then expanded in the presence of IL-4 and IL-7 in a gas-permeable culture device called a G-Rex. Next, after 7 days of culture, the cells are split and reseeded, and their media is replenished in order to sustain maximal in vitro CTL expansion. Finally, on day 12, cells are harvested and tested for purity, potency, and safety, and are cryopreserved for subsequent clinical use. Ultimately, CTL lines with multivirus specificity and minimal alloreactivity can be generated, as evidenced by their intracellular cytokine staining, interferon gamma production, and cytotoxicity.
The manufacture of multivirus-specific cytotoxic T-cell limes is time-consuming and cumbersome. It requires the use of Epstein-Barr virus transformed B-cell lines and adenoviral vectors, and the procedure takes about 3 months. Our new method does not use viral vectors. The cells grow exponentially in culture with minimal cell loss, and therefore the procedure is rapid and cost-effective. Demonstrating this procedure is Ulrike Gerdemann, who's a clinical fellow in the laboratory.
Harvest monocyte-derived DCs, which have been generated in CellGenix media supplemented with IL-4 and GMCS.